Hello, everybody. I'm Cindy Braden, the SVP of Sales for EnsoData, and I want to thank you for joining our webinar today. So with that, I'm going to hand it over to Chris Fernandez.
Thank you, Cindy, and very excited to share our updates with you all today. So we have an excellent panel with myself, Chris Fernandez, co-founder and CEO of EnsoData, Sam and Yoav on our AI team, and two of our wonderful collaborators, Breton and Dr. Nate Watson. So to dive right in, at EnsoData, we're passionate about research, and so far, our AI and machine learning research is really focused on diagnostics, treatment, and preventative care in sleep medicine, both around today's problems and the problems of tomorrow. We'd like to start today by sharing some of the key insights from a selection of our sleep research publications that align with three of the biggest challenges in sleep medicine today. First, the epidemic of undiagnosed sleep apnea. Second, the challenges in maintaining long-term Cpap adherence to support the best patient health outcomes. And finally, a little bit of a chicken or an egg problem, the clinical limitations of the AHI and strategies to go beyond the AHI. As Cindy mentioned, we'll conclude today with a fireside chat and an open forum Q&A with some of our research collaborators at leading provider organizations all around the United States and the world. So let's take a look at the first challenge. Undiagnosed sleep apnea remains an epidemic scale public health crisis today. Systemic inequities in access to care also persist in underserved ethno-racial communities, rural and critical access areas, and low-income communities. We pose the question to explore, can we utilize data that already exists today, is ubiquitous and widely available, to help clinicians achieve a greater scale of patient and provider engagement, education, and ultimately screening and diagnosis? We did this by looking at some of the most ubiquitous data that's available in healthcare today, data from the electronic health record. Some of our prior research, including our work on computational phenotyping in 2015, looked at our ability to combine this kind of EHR data with sleep study data to predict outcomes like 5-year all-cause mortality. This time, we looked at sleep apnea itself. And so to do this, we analyzed two different data sets, a 5,583 patient data set of type 2 studies that included 90 EHR variables based on the common clinical data set standard, and a second data set of 1,037 type 1 PSG studies with an additional 54 EHR CCDS-based variables. We used cross-validation to study this data using machine learning for 4 different outputs: normative sleep breathing, mild sleep apnea, moderate sleep apnea, and severe sleep apnea. And finally, what we studied here were comparisons involving 5 different sort of combinations or permutations of input data. First, just the gold standard stop-bang screening assessment itself, the Epworth Sleepiness Score, EHR variables, stop-bang, and EHR variables in combination, and finally, stop-bang with a random forest machine learning algorithm. So let's see what we found. The first the first result that we wanted to show is the comparison of using the stop-bang to predict mild, moderate, and severe sleep apnea versus the stop-bang plus some of this EHR data to do the same task. To measure that output performance, we used a receiver operating characteristic curve area under the curve, which is a robust statistical estimate of accuracy that's based directly on the sensitivity and the specificity for recognizing undiagnosed sleep apnea. So let's see what we found. First, we showed that combining this stop-bang data, the 8 questions from the stop-bang questionnaire, with some of this commonly available data from the electronic health record, improved screening performance. The ROC-AUC exceeded that of the traditional stop-bang when combining it with additional EHR data parameters at each of the mild, moderate, and severe thresholds. So this is not a super surprising result, but does show that more data is better and that we can take advantage of some of the data that already exists to provide more accurate screening. Second, we wanted to look at how the individual elements of that data, how valuable they were for being able to predict undiagnosed OSA. So to do that, we ran each of our 5 input variants through a decision tree, and we used the Gini index to calculate feature rankings of feature importances. So this graph on the right is a little bit of a explanatory graph for what the Gini index shows. On the left, perfect equality. If a variable is 100% correlated, it can have a Gini of 1, and if it's highly uncorrelated, a Gini of 0. And if it's highly uncorrelated, we can get a Gini coefficient that's close to 1. In machine learning, we want uncorrelated feature variables because that means they tend to be different in relation to the things that we want to predict in a consistent way. And thus, high Gini index means that we have informative and uncorrelated features that can be valuable. The random forest is an extension of this decision tree methodology that uses a technique called the ensemble method to combine multiple decision trees together into a single algorithm that are each trained on different data sets using bootstrap sampling. Random forest can output the same kinds of feature rankings as an individual decision tree using that Gini information. And so what we have here, the two tables on the right cover the SHHS data set for the traditional stop bang and EHR, and similarly on the right, the stop bang and the EHR for the second data set. What we found was, looking at the top 5 variables from each and their Gini coefficients, that the SHHS, all 5 Gini indices were greater than those from the SHHS data set from those EHR variables for predicting undiagnosed OSA. Similarly, in the second data set, 4 of the 5 stop bang variables ranked in the top 5 across the stop bang and the EHR data. So what does this mean? This means that the stop bang data is good. Generally the data that was included in the stop bang questionnaire were already some of the strongest predictors of undiagnosed sleep apnea. And so that prompted us to look at, one final analysis. And this was perhaps the most surprising, finding of all of this study. What we did here was we compared the ability for the traditional stop bang to predict that, sleep apnea area under the curve with the receiver operating characteristic. We compared that to the stop bang, the same 8 questions, the same input data, but run through a random forest instead of through the traditional calculation for turning that into a stop bang score. What we found was that utilizing a random forest with the stop bang, the same 8 variables that we use today, just a different calculation, allowed that method to exceed the stop bang in both data sets for all 3 of the sleep apnea disease state severity thresholds. What this indicates is pretty exciting. That that EHR data can help us to meet and exceed the stop bang. The stop bang features are good ones, but most interestingly, that there's really is low-hanging fruit. Given that the stop bang random forest exceeds with the same data, using even some of the most basic machine learning techniques to look for more sophisticated patterns than simply adding, subtracting, and multiplying allows us to capture improvements in sensitivity, specificity, and accuracy for the stop bang itself. And so, in conclusion, our big takeaways here were that the stop bang and EHR data can provide further insight into undiagnosed sleep apnea really in a new way at the population health scale. That stop bang is an effective sleep apnea screening tool that already uses many of the strongest OSA predictors. There's also still low-hanging fruit. Simple machine learning has been shown to elevate the stop bang performance beyond its traditional score. And really, AI can contribute to useful tools for sleep navigators to engage and to educate patients and other providers. When we think about some of the biggest challenges in operationalizing, these kinds of tools, the AI detection is really only one piece of it. And an even greater challenge is in the conversion and the navigation of patients, through education, ultimately through screening, testing, and diagnosis. And so, developing methodologies to increase patient and provider education and engagement that can be supported by these AI tools in sleep navigation represents a big opportunity. Similarly, future work also entails prospective validation of this approach for the application on claims data to take advantage of this population health OSA screening and predictive analytics for organizations that have more accessible claims data than EHR data. And finally, running a multi-site validation with the stop-bang random forest. This represents a pretty simple opportunity to get more accurate screening and severity categorization on every patient that we screen. And so that concludes the work that we wanted to highlight for the first challenge. I'll go ahead and jump right into number 2. And so the second challenge we wanted to talk about was the challenges with maintaining long-term Cpap adherence to support the best health outcomes. Maintaining adherence and compliance with a positive patient experience remains a major challenge today. Some sources have cited 12-month compliance on Cpap, as low as 1/3 by the end of first year in some studies. A growing body of evidence also supports the trend that sleep clinician managed patient adherence, including outreach, follow-up, and behavioral coaching, can achieve substantially greater results in terms of 12-month compliance. So in this work, we posed and explored the question, can AI be utilized to support clinicians in improving and maintaining long-term adherence with accurate predictive results instead of reactive information? Our goal in doing so is to enable a more systematic, proactive, and preventative approach to supporting patients who may be struggling the most on Cpap therapy and understanding behavioral methodologies to make that as successful as possible. So, first I've pulled up, some Cpap PAP usage data on the top days per month where the patient used their Cpap 0 hours, less than 4 hours in the middle, and greater than 4 hours on the bottom. The y-axis here is the last month, the previous month, and the x-axis is the current month. So when we plot Cpap usage on a month-over-month basis, we're able to see that Cpap adherence is highly variable. Month-over-month usage, does tend to change constantly. It does have momentum, but the swings up and down can be quite large. And so, this contributes to some of the complexity in, knowing how to best prioritize resources. So to study this question, we utilized two input data sets. One with 14,000 patients, at least 450 days of PAP therapy usage data per patient, which included more than 6.3 million nights of Cpap data in total. We also used a second data set to confirm that we had good site-to-site test-retest reliability and reproducibility with these methods. In terms of outputs, we utilize this input data to look at 3 different adherence monitoring categories, either with regression coefficients or with sensitivity, specificity, and accuracy. Those outputs were a predictive measure of PAP usage in days, a predictive measure of PAP adherence group, identifying patients to between 3 and 4 clusters to define those groups, and finally, predictive PAP compliance using a binary interpretation of the CMS criteria. In terms of comparisons, in this work we compared 2 different machine learning variants, supervised learning for one and unsupervised learning for the second. We'll talk a little bit more about what those are, but this included an evaluation of clustering, regression, and classification to achieve those adherence monitoring outputs. So, in terms of sort of the algorithmic architecture of this AI model, the basic setup was we feed in this historical trailing PAP usage data. We can also feed in additional demographic or other metadata parameters. This is run through a deep neural network, AI, and machine learning algorithm. And that is iteratively optimized such that over time, it improves performance as it gains experience and sees additional data to be able to accurately forecast the usage in days as well as these different adherence groups. And so let's look at our first takeaway. What we have here are a visualization of kind of the supervised machine learning setup. What we found here was that supervised machine learning can be used to configure, essentially to design, as well as to accurately predict PAP adherence groups. The graph on the left shows an example of how one is able to kind of configure these kinds of supervised groups that an algorithm can then be trained to recognize and that are informed by clinical judgment and expertise. So we colorized those different combinations of usage using a rating scale, and ultimately designed and proposed 4 different groups that patients would fall into in different combinations of those ratings. We were able to take advantage of this with supervised learning and achieve accurate performance for classifying the future usage and the future group. What does that look like? Well, it looks a little bit like this. This is the same data that we looked at at the beginning of this section on the left. Given that this is a plot where we have the last month versus the current month, if everything was the same month over month, we would simply have a diagonal line down the XY axis with a slope of 1. In contrast, if things were completely uncorrelated, we would have something that looks more like a square. And that's basically what we have. What the AI was able to do was to achieve substantially greater agreement and strength of fit for the next month of usage and the next month of groups compared to a model that would take as an assumption things are probably going to be similar next month. So we observed R-squared values for that regression of 0.94, 0.87, and 0.95 for each of these different thresholds specifically with our AI model. Yep. And so next, we decided to try a little bit of a new technique. This has become a famous cake reference by Jan LeCun, who is the current VP and Chief Artificial Intelligence Scientist at Facebook. He compared, reinforcement learning, supervised learning, and unsupervised learning in the following analogy. If machine learning was a cake, reinforcement learning would be the cherry on top, supervised learning would be the icing, and unsupervised learning would be the cake. Unsupervised learning provides us with a way to study data and relationships between different examples of that data without imbuing our preconceived notions or any a priori information that might bias the results that we see. That makes it a more challenging learning process as it's more open-ended and more general. However, this can be a very powerful approach in combination with supervised learning, or in some cases by itself. So we wanted to see how an unsupervised learning approach would be different relative to the supervised learning approach we just looked at. And so what our big takeaway from that analysis was that unsupervised machine learning can learn adherence patterns without predefined groups. And so, the way that we did that was initially by clustering the data, the unlabeled Cpap usage data, to identify, 3 different clusters that had a very high degree of internal consistency within a cluster and statistical differences between clusters, and utilized the clusters that this unsupervised machine learning approach learned then for prediction, instead of those that we had predefined ourselves with the rating scale and with the 4 prior groups. The big takeaway here is that we were able to achieve accurate performance both with predefined or with unsupervised learned groups that could accurately predict and forecast Cpap usage. We've already looked at some of the R-square regression values, and in terms of next month classification, based on these different groups, we achieved a 93% sensitivity, 96% specificity, and 96% accuracy for the supervised setting, and a 95% sensitivity, 96% specificity, and 96% accuracy for the unsupervised learning setting. And so, our conclusions from this study were the following. That one, supervised AI can enable really customizable definitions of adherence groups for predictive research and for clinical studies. That unsupervised AI can help us find useful and general relationships in adherence data that can be hard to see. That both AI methods were shown accurate to forecast future PAP usage, future PAP groups, and even behavioral factors. And that novel outreach methodologies will be needed and can be studied to develop clinical best practices for utilizing predictive instead of reactive PAP monitoring tools. Some of our future work for this particular project involves the incorporation of additional data sources like the EHR, waveforms, and others, and critically to validate the relationship between Cpap adherence and hard outcomes. This includes data like future and historical episodes of stroke, acute care utilization, and other metrics. Additionally, as mentioned, and similar to kind of the adherence problem, the ability for AI to accurately detect these kinds of scenarios is really only one part of the problem, and solution. And in our view, the even greater challenge is really developing and optimizing new patient engagement methodologies that can translate into optimized and sustained behavior change, greater self-care and responsibility, and kind of maintenance of those health outcomes over time. For this, we suggest a randomized, blind, placebo-controlled study, in part to study if there may be any differences that manifest behaviorally through the Hawthorne effect if patients were to know they have predictive instead of reactive support. And finally, greater opportunities to utilize the AI to assist clinicians in identifying alternative therapies for patients who are failing or struggling on PAP therapy. Examples of this include neuromodulation therapies, which are continuing to grow smaller in size and finer in their electrical and modulatory control, and also treatment regimes for frequently overlooked comorbid conditions like OSA and insomnia together, which could negatively compound a patient's OSA therapy if not addressed as well. We have a forthcoming publication on neuromodulation to this end, and we'll be excited to share that. So those were our big takeaways for number 2. Now, in the kind of final 5 to 10 minutes of this talk, we will conclude with challenge number 3, the chicken or the egg problem, clinical limitations of the AHI and strategies to go beyond the AHI. So today, there is growing awareness of the limitations of the AHI. Recent studies have not only challenged the quality of evidence and the standard of evidence for Cpap therapeutic outcomes, but also the AHI and its ability to correlate well with various adverse health outcomes, costs, and care utilization types. We know that 90-second desaturations are probably not equal to 10-second desaturations. 15% is likely worse than 3%. That sleep likely occurs in more than 5 states, maybe even multiple states at the same time. And supporting that, there is a growing body of clinical and scientific literature that is currently aiming to develop, validate, and understand potential indices and metrics that can take us beyond the AHI. And in doing so, tell us far more about possible patient subtypes of OSA that provide stronger predictors of adverse outcomes, costs, and likely optimal therapeutic pathways and parameters. Things like if a patient has a lower or high arousal threshold, does that impact the sleep fragmentation that might manifest with an equivalent therapeutic pressure on a device like a CPAP. So all that said, this presents a chicken or an egg problem in that the current standard of care is based entirely on the AHI. Thus, novel indices that can take us beyond the AHI must presently be compared to and validated in comparison to the AHI. This is a solvable problem, but resolving when differences from new metrics or indices relative to the AHI are better or worse differences, desirable or undesirable differences, remains an open and, as of today, a unsolved challenge. And so the first study that we did to explore this was a study of brain age. So sleep apnea is understood to impact neurocognitive performance and outcomes. Early work in sleep apnea subtyping and phenotyping has suggested that this neurocognitive effect is not uniform, is not exactly the same in all patients. Some prior researchers have hypothesized that the long-term effects of undiagnosed sleep apnea may manifest in structural and functional brain changes that are observed to present similar characteristics to advanced or accelerated aging. Prior work has demonstrated relationships between a computationally derived brain age value that is measured through EEG data instead of the patient's chronologic age. And additionally, some researchers have hypothesized that anomalies and deviations in a patient's observed brain age may also be an early predictor for neurodegenerative diseases like Alzheimer's and Parkinson's, as well as neurodevelopmental conditions in their progression. As such, we developed, tested, and evaluated a brain age model using a novel AI and machine learning phenotyping approach applied to the largest EEG PSG data set tested and published for this application so far to the best of our knowledge. That data set included 126,241 PSG studies for training, 1,172 for initial testing, and finally two 100-patient semi-prospective validation data sets. So let's see the results. The basic setup for the AI model was similar to that of the Cpap model. Input included neurologic data from EEG and EOG. The model was trained using a deep convolutional neural network, which would predict an age based off the patient's brain data, would compare that to their chronologic age, and would iteratively repeat this to optimize performance and generalization. So what did we find? The first takeaway was that our brain age model demonstrated state-of-the-art performance with a mean absolute error of 4.61 years, less than 5 years. So on average, the brain age index could get within 5 years of the patient's true chronologic age simply based on characteristics it could detect in the EEG data. We also observed strong regression performance with this, with a slope value of 1.076 and an associated R value that was greater than 0.90. And so let's unpack this a little bit. First, we took that brain age and we translated it into two different indices. The first was a brain age index. The brain age index distributions showed consistent differences in multiple conditions. And so the brain age index is simply the AI's predicted brain age based off of that EEG. You can see the condition positive in orange and the condition negative in green groups for depression, diabetes, and hypertension here. And you can observe visually that there are some subtle differences in the distributional characteristics like the mean and the standard deviation of the brain age that was observed in patients who are condition positive for conditions like depression or diabetes, or were condition negative. What this showed was that, brain age demonstrated consistent differences for multiple conditions, such that in effect, it appeared that it could almost predict multiple conditions. The second takeaway was related to the absolute brain age index. And so these look a little bit different that you see on the bottom. The absolute brain age index is the absolute value of that brain age, and it's the difference relative to the chronologic age. And so really what this showed us were well, the brain age index from the prior slide could be greater or lower, on average based on the presence or absence of a condition. For this one, it was really about the size or the magnitude of the differences or the deviations. So absolute brain age index for seizure disorders, stroke, and severe arousal index demonstrated significant distributional characteristics whereby the brain age deviation was a lot larger, in the presence of those conditions. Furthermore, significant differences resulted, as we mentioned, in the directionality. Is it greater or less than for the brain age and the deviation? How big is it for the absolute brain age? For the brain age, for diabetes, hypertension, depression, low sleep efficiency, Epworth Sleepiness Scale, memory and concentration, and for the absolute brain age in low sleep efficiency, severe arousal index, stroke, seizure disorders, and a severe desaturation index. We also utilized the regression analysis to analyze the effect size and to rank the effect size that brain age and absolute brain age index deviations had by condition. And so here is the last data that we have for today. First, I think y'all are already very familiar, but this work kind of focused on a sensor called the PPG, the photoplethysmogram. And so this is a sensor that's used ubiquitously in home sleep apnea testing, as well as in consumer sleep tracking wearables. Home sleep testing has been rapidly growing in adoption, since its inception, and has accelerated over the past 15 years. And with COVID, the AHI calculated from sleep tests is a fraction that is denominated by the total sleep time, which is currently determined by the EEG, under the gold standard of care. In HSTs, determination of epoch by epoch sleep-wake staging remains a major challenge for human expert visual pattern recognition, as well as for, potentially antiquated HST auto-staging systems and approaches. So here we pose the question to explore, if AI could contribute to helping to improve the diagnostic reliability and accuracy of home sleep testing using a smart AI transfer learning-based approach to assessing sleep stages and total sleep time in the absence of EEG. So to do this, we utilize a novel single-channel photoplethysmogram approach, to explore how accurately AI and machine learning could detect sleep based on cardiopulmonary data, and compared that to the gold standard PSG EEG based sleep staging that was analyzed by a panel of sleep experts. And so kind of our basic hypothesis for this was there's a lot of data that we can get out of the photoplethysmogram. Currently, there are existing algorithms to calculate respiratory rate, pulse rate and heart rate, and blood oxygen desaturation levels. And one might hypothesize that variability in those variables, and potentially others in the raw PPG, can provide enough information to detect, sleep versus wake with high accuracy. So to test this out, we used 4 different data sets, a little bit more than 11,000 patients for training, 2 different test data sets at 1,200 and 17,000, and then finally a clinical validation data set that included 100 adult subjects. We analyzed 2 different outputs, specifically the epoch by epoch sleep stage accuracy that was based from the PPG, and also the total sleep time index that was based from the PPG using ROC, Bland-Altman, and Deming regression analysis. We facilitated these comparisons from the PPG-based AI scoring of sleep stages in TST to 2/3 majority scoring of EEG and to AI scoring of EEG for the same outputs. And so we had a few quick takeaways from this. First, PPG-based total sleep time showed consistent performance across multiple devices. So the verification initial testing for this involved more than 20,000 studies from many different devices, more than 10 different devices. And in this example, we have on the left PPG total sleep time, on the right EEG total sleep time from patients who had a simultaneously recorded EEG and PPG in the context of their PSG study. And you can visually observe, the high degree of consistency and concordance between both of these separately calculated total sleep times. Second, that PPG sleep staging, the epic by epic sleep stage determination of sleep or wake, demonstrated strong sensitivity and specificity with a receiver operating area under the curve of 0.96 in comparison to the ROC for the AI for EEG, which was all the way up at 0.99. And finally, utilizing the epic by epic staging output from the prior experiment, we ran a final analysis using the Bland-Altman analysis of differences, comparing that PPG total sleep time then to the panel scoring. We observed here that the PPG total sleep time had a mean difference of less than 6 minutes over the 100 patient sample in comparison to manual scoring, and clinically acceptable, LOA, relatively tight limits of agreement that exceeded, much of what we had observed in prior publications. And so to conclude here, brain age and absolute brain age index are, very, very interesting variables that hold promise to be able to tell us a little bit more about sort of how age tends to change different physiologic systems, to be able to characterize those age-related changes, and to be able to detect deviations, in those changes that may arise from pathology. That brain age and absolute brain age index were well correlated with several health variables that were previously hypothesized to be affected by accelerated aging and neurocognitive impacts. And finally, that a reliable and accurate total sleep time and sleep staging could be estimated by AI from a single channel PPG, a single channel photoplethysmogram. And so I think our big takeaway here is that there's a big opportunity for clinicians to use AI as a tool to study, define, and discover novel biomarkers and OSA phenotypes that go beyond human perception and visual pattern recognition, and have the potential to take us beyond the AHI, closer to personalized and precision medicine. We don't necessarily, we don't propose that the brain age is sort of the new AHI, but more so that the experimental framework and the AI methods that we use to derive the brain age are applicable in a very general way to the larger problem of understanding OSA subtypes. And so future work involves the extension of this PPG staging to include additional outputs for REM, deep sleep, and consumer sleep tracking wearables, to study the more dynamic characteristics of brain age with a multi-night evaluation study. How does brain age vary from night to night? Does brain age change in response to Cpap treatment or other treatments for OSA? Over a longer time scale, how fast does brain age change in relation to age? And finally, getting into the next level with OSA subtypes that are really centrally based on health outcomes, comorbid diagnoses, and optimal therapeutic pathways, and can take a approach in their design that they can truly identify data model and method independent phenotypes, such that you can find those phenotype groups in any cohort of patients from any location using any method, whether or not that involves AI. And so those, that's sort of my conclusion. I'd love to jump into kind of the fireside chat. We are greatly appreciate you joining us today to kind of discuss this research. And so we would love to hear what questions that you have and to engage on those. Thank you.
Thanks, Chris. I can dive into the questions too, but I wanted to give an opportunity for any of the panelists if there was anything they wanted to add. Dr. Watson, I wasn't sure if you had any comments before we jumped into the Q&A.
I do, and thanks, Chris, for such a great presentation, and it's really compelling, the work that you all are doing. I mean, I have lots of comments to make in general, but maybe I'll, for the moment, focus it on brain age metrics and measurements. And, it just strikes me how we know that sleep impacts all aspects of human physiology. And so and then in turn, the brain is impacted by all aspects of human pathophysiology. And I think that's what we see in these results. And you can actually close the loop because we know, for instance, obstructive sleep apnea causes hypertension. And now we also know that hypertension can increase brain age. And so, we always tiptoe around causality, and I think we still have to be cautious. But, what we see in the data here is that, reduced sleep efficiency and increased arousal index, being associated with increased brain age, would seem to kind of suggest that either through sleep apnea or other elements that the problematic sleep itself is kind of damaging the brain over time. So, to me, that it's just such an interesting finding, and in a sense, we may be moving towards what I like to consider a vital sign for the brain. And of course, the challenge will be gathering EEG on a population scale, and that's a big challenge that would need to be tackled. But I think that, to me, from a preliminary comment, that's the most one of the most interesting things that Chris discussed today.
Excellent point. Breton, did you have anything you wanted to add, or you guys want me to jump into the questions that came up?
Oh, yeah, let's just jump into the questions.
Okay, perfect. So hopefully everybody has had a chance to get their questions typed in. We do have, gosh, a whole 15 minutes here. So if anybody had any other ideas, go ahead and toss them in and I'll get them asked of our panel. The first question that came up was, what are the potential impacts of stop bang random forest combination, perhaps in underserved communities? Dr. Watson, I don't know if you want to take that one, or does any?
Yeah, sure. I think hopefully what this does is, this can be integrated with medical records in order to provide kind of an indicator to primary care doctors that they need to consider a sleep disorder or sleep apnea as a potential problematic factor in the health and well-being of the patient that's sitting in front of them. And we know right now, unfortunately, sleep medicine is really underdiagnosed and underappreciated by primary care providers. And it's not necessarily their fault. It's just undertaught in medical schools and residencies around the country. So I think, having the random forest plus the stop bang and integrating that into the medical record might really provide opportunities to kind of trigger a conversation between a primary care doctor and a patient in order to begin to discuss their sleep issues, to hopefully motivate a referral to a board certified sleep doctor to get the issue addressed.
Very good point. And I think it actually leads into the next question, which is more focused on the Cpap research. So Breton, I think this one might be more directed at you, but speaking about the patients, the next question is, what is the consumer effect of the Cpap research? In other words, why would the patient population care about this research?
Well, I think that the patient population will care about the research just in the fact that it's going to ensure that they'll be more successful on their therapy. They spend a lot of money, being compliant, getting diagnosed, tested, and becoming compliant. So this type of research will help them. I think also we'll be able to figure out, for example, the effect of different modalities, which we transfer information, whether it's educational or motivational, and find out how it affects behavior change within especially the variable group. And so I think that, for example, we could use text and app messaging as well as via phone. And I think that the consumers would appreciate being messaged in a variety of ways that were effective.
Excellent. The next question that came in, I'm not entirely sure who wants to take a stab at this one, but it says you talked a lot about OSA, but beyond that, what else could AI assist with? So I'm assuming it's specific maybe to this research, or? just beyond this research in general. Chris, do you want to take a stab at that, or maybe Sam or Yoav?
Yeah. I think there's two really interesting directions. One of the themes that I think that felt like a pattern in that last set of challenges was the ability for AI to detect patterns that can be very counterintuitive or even very difficult for us to see with our eyes in kind of a simple way. And so I think that some of the places where that type of analysis can be the most useful and advantageous is when we're dealing with really complex data. And so, as Nate mentioned earlier, I think there's a really interesting opportunity with kind of EEG and brain-based diagnostic and waveform testing, particularly in the home setting and over longitudinal timeframes. And I think similarly, there's a lot of exciting work happening in the space of consumer sleep tracking. Now everything from smartwatches to I guess even our thermostats are looking to get involved with sleep tracking. And so I think there are also exciting opportunities for patients with chronic cardiopulmonary diseases that could be detected in very interesting ways using that kind of cardiopulmonary setup.
Let me jump in here as well. There's a very interesting study that came out recently from Dr. Mignot's group at Stanford, where they looked at patients with type 1 narcolepsy, and they generated what they called a hypnodensity graph of sleep stages, or really a probability distribution of sleep stages. And using this approach, they had a very high sensitivity and specificity for differentiating type 1 narcolepsy from a control group. So I think, the application of AI to sleep medicine is definitely going to reach beyond the borders of sleep disorder breathing as we continue to experiment and learn and advance in this area.
Excellent point. There was a question, and Yoav, this one might be a little more directed at you. And Chris, I think you discussed it a bit with your next steps, but there was a specific question on what is actually planned for future research on these topics. Chris, I know you mentioned a couple of things that would be worth looking into, but Yoav, perhaps we can ask you, is there anything planned currently to do any of that follow-up research?
Yeah, well, I mean, we, for example, for the brain age, we looked at a variety of different variables: hypertension, depression, diabetes. It will absolutely be, very interesting to add more variables to the list of variables we already looked at, mostly neurodegenerative diseases, Alzheimer's, Parkinson's, and so on. So that is definitely, a next step. Let's see what else. For example, for the the PPG signal as well, we saw that we can get the respiratory rate out of the PPG, the oxygen saturation from the PPG. Now also sleep versus wake. Can we also get the more specific the specific differences between the the different sleep stages? And what else? So the thing is, we're dealing with a lot of highly dense data that contains a lot of information that is recorded for 6, 7, 8 hours a night. So what else can we extract from those signals, and basically what can we use our AI and what can our AI detect from those signals that humans can't, right? So it's not only trying to simplify the tasks that humans already do, but also use and leverage AI to try and find structures that the naked eye can't. So all of those topics are for sure a part of our research roadmap.
That's excellent. Okay, we have one more question. Oh, somebody just typed one in. I'll have to read it. We had one more question. Looks like we have two now. But this next one, my goodness, I know, Dr. Watson, you would be able to answer it, but it might be something that the entire panel, you might have thoughts on it. But kind of leading to the question that was just asked, the next one is, what's next for AI, particularly in sleep medicine, or what's your dream outcome as a result of it? Dr. Watson, do you want to take a pass at that? And then if anybody else has any other comments.
Wow, that's a great question. I mean, what's the dream? The dream is to put myself out of business and have all the sleep disease in the world taken care of, and you know, that's obviously quite lofty. But I think, you know, when I look at this, you know, when we look at the PAP adherence data, for instance, what I'm interested in doing and learning is swimming further upstream. You know, let's get these cluster analysis done and identify these non-adherent clusters, perhaps before we've even tried them on PAP therapy. I mean, if we can identify who these individuals are that early, you know, we can get them onto a more appropriate, effective treatment faster. So we reduce suffering and we reduce cost that way. And so that's, you know, Chris mentioned personalized precision medicine. I think, you know, that's what the ideal would be to really realize what that looks like, which is essentially getting people on the best, most effective treatments as soon as possible.
Excellent. Does anybody else have any other thoughts?
Yeah, Cindy, I can add a couple, and I think Nate, in terms of, yeah, I think I absolutely agree in terms of the long term of how, you know, we think about really using algorithms as a baseline support for triaging patients through diagnosis and treatment. I think though, even in the next, you know, 3 to 5 years, I think it's clear that AI should be a tool that all researchers and investigators are using as kind of a core tool set to investigate these diseases. You know, given the fact that these algorithms can do things that humans find it difficult to perceive with their own eyes and cognition, you know, we need to use this as kind of the mechanisms to explore the diseases and sometimes even work backwards to say, hey, the model can predict this. What does this mean physiologically? What might be happening on the mechanistic level so that we can understand better? And I think that is a tool set, and I think something that we're going to hopefully see a lot more researchers and doctors using as a main microscope to look at these diseases through.
Gets me so excited. I get caught up in all the answers. Okay, we did get 2 questions specific to pediatrics. So first one, and I'll ask them both together because I think pediatrics, I think they're tied together. First one is, did you do any of these studies using children or pediatric research? And then the follow-up question is, many pediatric sleep labs are afraid to jumpstart HST. Do you have any data that encourages them to utilize HST? I'm not entirely sure. I'm guessing that's either Breton or Dr. Watson.
Dr. Watson, I'll let you take it. Ours, we don't have the pediatric population.
Yeah, and Chris can answer as well. I'm not aware that they've done work in pediatrics, but it's possible that some of this would relate. And then, you know, from a home sleep apnea testing perspective, I think in children, the COVID pandemic may have changed the calculus on that a little bit, because in discussing this with my pediatric colleagues, you know, obviously when labs shut down for obvious reasons, you know, I think the pediatric sleep community began to realize that maybe we need to begin to look at home sleep apnea testing, at least in their teenage patients, in order to get a diagnosis and get them on treatment during the pandemic. So, you know, I'm not a pediatric sleep specialist, so I won't answer it beyond that, but I think that the pandemic has changed a lot of things in sleep medicine, and this is one of them. I think that pediatricians are appreciating that home sleep apnea testing, at least in their adolescent patients, is something that can be applied.
Yeah, and I think, you know, if we'd be happy to share that information as a follow-up to the question asker if interested. We did do an N=100 clinical validation study on pediatric cohort as well, semi-prospectively, that included total sleep time and brain age. The results were very interesting, as one can imagine. You know, the difference of 8 years between, or 7 years between 1 and 8 is a lot different than, you know, 81 and 88. And so we really had to optimize for a greater level of, the sensitivity of the algorithm to calibrate it into that tighter range. So we did, tend to see longer total sleep times and sleep stages that were more resilient to fragmentation in pediatric patients, and overall, performance that was highly consistent, in some cases even better than in the adult population.
Great point, Chris. All right, we have one last, so this is going to be last call if anybody else has any other thoughts. That's a bit of a question / comment, perhaps more of a comment, but I'll see if anybody has any follow-up that they want to offer to it. But basically, to Dr. Watson's point that he was just making, the concept that really having a focus on providing some sort of predictors on which therapy might be appropriate for each patient, and if AI would have the ability, to do something like that. So I don't know if I would say absolutely everybody agrees with that. I don't know if anybody has any other thoughts on it.
I think that it could, as we drop more variables in, and looking at claims data, disease severity, and going towards adherence and compliance in the future. But I definitely think within the variable phenotype, there's probably two levels. There's a variable that's moving in a positive direction and a variable that's moving in a negative direction. And the sooner we can see people from their onset of therapy, maybe PAP therapy in this case, onset of PAP therapy, that they're headed in the negative direction and they're on a more negative variable state, intervening more quickly while they're still interested in getting treated and they're not dissuaded by too long of a fight.
Such a great point. All right, folks, that was the last question that came in today. Chris, I'll let you take it home, and then I will just personally say thank you to everybody for attending, and then obviously a huge thank you to our panel for all of the work with the research and then joining us today.
Yes, thank you very much, Cindy, and thank you again, wonderful collaborators, for joining today as well. Really enjoyed the conversation, and for those that joined us to listen, we'd love to hear your questions. So please reach out. This is very interesting stuff, and we'd love to dive in on it even more if you'd like to chat. So thank you again for joining, and hope you have a lovely rest of the week.